Okay, so as we previously said, insulin, glucagon, cortisol, and epinephrine are our major carbohydrate metabolism hormones, or the major hormones involved in carbohydrate metabolism. So, as we know, insulin is important in lowering our blood glucose. So, what we need to realize is that insulin isn't just lowering our blood sugar, it's telling our cells to take up glucose, right? So, there's all this free blood glucose running around in our blood, and insulin tells our cells to actually take it to be used. So you'll see that it has an anabolic effect on carbohydrates. So it can either be anabolic, catabolic. Anabolic means it's building. Catabolic means it's breaking down or destroying, right? So we call it an anabolic effect on carbohydrate because whenever we have an excess amount of glucose in our blood that our body isn't going to completely use to create energy, we're going to store some of that, right? We're going to store it as probably glycogen or as fat. So that's why we say it has an anabolic effect. Of course, we know insulin's produced by the beta cells in our pancreas, and we are able to produce about 40 to 50 units of insulin per day. So think about that the next time you talk to someone who's on insulin for their diabetes, and just kind of compare that to what you should be making on a normal basis. So not only does insulin increase uptake of glucose, it also kind of, or does in a small fashion, increase the uptake of amino acids. So the presence of insulin is going to start all of these metabolic processes, right? So it's going to help us to break down blood glucose. It's going to help with the pentose phosphate pathway. It's going to help with glycogenesis. Like we said here, we're making glycogen because we have this excess glucose hanging out. And as well as lipogenesis. We're going to create fat because we have all of this excess glucose. Let's see, and here you see it's going to inhibit glucose formation. So that means it's going to inhibit glucose formation from non-carbohydrate sources. So gluconeogenesis here. If there's insulin or high amounts of insulin running around in our blood, we're not going to be putting our cells through gluconeogenesis as readily because we're going to have all of these carbohydrate stores being created, and if we did need it, we would be able to pull from those. So again, we're inhibiting gluconeogenesis. We're inhibiting the breakdown of fat because if there's a lot of insulin, that means we have excess glucose, so we're going to build fat, right? So we're going to promote the storage of energy in either glycogen or fat storage, and then promote the synthesis of proteins, right? Just because of its role in initiating other metabolic processes. Okay, so make sure that you know these metabolic roles. All right, so glucagon, we know it's created in the alpha cells of our pancreas. Glucagon, remember, if glucose is gone, glucagon's going to act, right? So its role primarily is to increase blood glucose, right? So it's going to, one, promote the conversion of non-carbohydrate molecules to glucose, so it can influence gluconeogenesis. It's going to promote lipolysis, so it's going to want to break down fat to be able to put that into gluconeogenesis to create more glucose. So as you can see, glucose is gone. We have two routes here that we're going to make more, okay? So you'll see that it promotes gluconeogenesis. It will break down glycogen here. You see glycogenolytic. It's ketogenic, right? Ketosis. And then lipolytic, all right? So it's going to break down fat, okay? Those are pretty easy to remember. So remember, glucose is gone. All right, and then we have epinephrine that we're going to talk about, as well as cortisol. These are our adrenal hormones, and you may remember them from, learning about the flight or fight response. So first, epinephrine, that's produced in our adrenal medulla. It's the most important substance related to our stress response. What it does is it increases the force and rate of our heart contractions. So what the purpose of epinephrine is for us to have a lot of blood flow to be able to move those muscles and get out of trouble, right? So to be able to do that, we need, this quick energy available, right? So we're going to start breaking down glycogen and we're going to break down fat to be able to create more glucose, right? So gluconeogenesis here, so that it can be used as quick energy. Let's see. And then again, cortisol, it's going to be in our adrenal cortex, or produced in our adrenal cortex rather than the medulla. And again, it's associated with stress, but also with fasting, okay? It has an anabolic effect in the liver. So remember, anabolic versus catabolic. So we'll put that in our back pocket. It's going to preserve circulating glucose, right? So if we're preserving glucose, we're probably going to store it as glycogen in our liver, right? So it's going to decrease glucose and amino acid uptake in our peripheral tissue. So cortisol's, it's kind of hanging out in there. It's like it knows our body's under stress and it doesn't know how long that stress is going to last. So it wants to help our cells kind of function on the down low as long as possible, okay? So it's going to make sure that our cells aren't using too much too much of the sugar that's running around, and it's going to save some, for in case things get worse. Okay, so you'll see some of these are a little contradicting, okay, because it knows we're under stress, but it also doesn't want us to go kaput, right? So we can, with cortisol, promote gluconeogenesis. So we're taking fat and we're taking protein and we're creating glucose, right? And it also promotes glycogen synthesis. So it's kind of funny, we're creating glucose from non-carbohydrate sources for energy, but then here we're taking glucose and we're storing it for later, right? We're making glycogen. All right. So remember, that's that check and balance. It knows our body's under stress, but it's not sure how long it's going to last, right? So it wants to make sure that we're going to be able to survive that. Let's see. Again, we're going to promote lipolysis because we're going to put that into gluconeogenesis. And, consequentially, it'll increase total white blood cell circulation. But don't worry about that too much. Okay, so as you can see, our blood glucose levels are highly regulated by our hormones, which those 4 that we talked about, right? Those hormones can affect, when we're going to be uptaking glucose and how rapidly we're going to uptake it, where it's going to be uptake. But we also have, 2 other ways that glucose can be regulated. Our enzymes can function a little bit differently. So don't get stuck in the mud with these. And then we have these allosteric influences. So basically, substances other than glucose can be kind of place holding those receptors, right? So our hormones are going to be the biggest influence on our glucose regulation, but we also have these other 2 means that are very necessary as well. All right, so we've talked about these different pathways. So just to review them again, glycolysis, we're taking glucose and we're creating 2-pyruvates for energy, right? Remember, glycolysis is the only one not happening in the mitochondria, and it's kind of the kickstarter for the rest of these metabolic processes. So this right here is kind of the steps of glycolysis if you want a better look at that. All right, this is glycogen synthesis. Basically, with glycogen, whenever we're creating that, we have this surplus of glucose. We don't need all that energy right now, so we're going to save it for later, okay? So with glycogen synthesis, we have this UDP glucose that utilizes glycogen synthase, okay? Glycogen synthase Remember, synthesis, you're building or creating. ACE is an enzyme, okay? So if you see glycogen synthase, you know you're building glycogen, okay? That's the enzyme that helps build glycogen. So if you see either glycogen synthase or UDP glucose, I want you to think glycogen synthase or glycogenesis. All right, and then we have the opposite, which is glycogenolysis, okay? So obviously here we're breaking down glycogen, okay? Not anything huge I want you to know about the process of glycogen degradation, other than the phosphorylases are important. Phosphorylases are going to come up later in this class, so kind of put that in your back pocket, all right? We have to use phosphorylases to liberate energy, all right? And whenever we create glycogen, you see we're using this UDP glucose, right? That has phosphate attached to it. So whenever we're creating glycogen, we're sort of locking it into our hepatocytes with this phosphate, okay? With glycogenolysis, we're using phosphorylases, so this phosphate enzyme, phosphorylase, to sort of take off that that attached phosphorus that's locking that glycogen in the hepatocyte, and that's what's going to free up the energy. So remember that. That's important. All right, pentose phosphate pathway. We're not going to talk a lot about this in this class just because there's more relevant pathways, but just know that it's there. Krebs cycle, we're going to get a lot of this. You've already talked about it a little bit in this class. There's about 8 reactions that are sequential, and it's all kickstarted by acetyl coenzyme A. NADH and FADH2 are important factors of the Krebs cycle that are going to be able to be used in the electron transport chain. The electron transport chain is like the most efficient means of creating energy. So it's really important that you understand that the Krebs cycle kind of has a turn in that. Okay, and then gluconeogenesis, like I've said before, is the creation of glucose from non-carbohydrate substrates. So it's going to happen mainly in the liver, and it's going to typically use, your fat sources or your protein sources. We don't want gluconeogenesis to be happening all the time. We want to be able to feed our bodies so that, our body's not using muscle stores or fat reserves that keep us healthy, right? So you'll see in patients in the hospital, malnutrition is such a big deal because they may not be fed properly or given enough protein or given enough carbohydrates to keep their body from using those protein stores and fat stores as carbohydrate sources. Okay, I believe this is another one of the clinical insights. So carbohydrate response element binding proteins, a mouthful. Just know that this can be activated with a high carbohydrate diet, and what it's going to do is stimulate fat synthesis, right? So like we've talked about before, if we have excess glucose hanging out in our blood all the time, our body's going to want to reserve it for a rainy day, right? We can reserve it as glycogen, or we can reserve it as fat in our fat cells. So just know that this binding protein helps to stimulate fat synthesis, and that it's found typically in a high carbohydrate diet. So you could venture to say that this is common, among people with a Western diet. Okay, hope this helped. If I was trying to keep it as short as possible so you wouldn't have to be watching a video for an hour and a half. So give me some feedback if you like this or not, and we can move on from there. All right.